Alright, so in this video we're going to talk about making riboprobes and synthesizing riboprobes. The protocol we're going to use is in vitro synthesis of riboprobes. And in other videos, and you may have already worked on this yourself, we talked about how in our lab we often clone genes into this PJC53.2 plasmid. So this is a gene of interest. You might have already done this yourself, cloning genes into this plasmid. And then elsewhere we've also talked about amplifying a gene of interest using a hot start PCR. And this PCR product has this T7, SB6, T3, and T7 RNA polymerase promoters on each side. And the two things we can do with this PCR product, we can actually use the same template, are to make double-stranded RNA using this T7 promoter and T7 polymerase, or we can make a single strand of RNA using either the SP6 promoter and the SP6 enzyme, or the T3 promoter and the T3 polymerase. And if we do this, then we get a single strand of RNA, and one of these strands would be the reverse complement and exactly bind to and match an mRNA, and the other one would be just, it would look similar to the mRNA. And so it wouldn't bind to the mRNA because they would be exactly alike, not a reverse complement. And so whenever you want to make riboprobes, one of the things you need to know is which of these enzymes, T3 or SP6, is going to give you an antisense sequence, because the antisense is what we want when we are making a riboprobe to bind an mRNA so that we can look at the gene's expression. So for today, I have a series of reactions to set up. I have already gotten some of the reagents pulled out. The first thing I have is I have my purified PCR products. So I have a set of tubes that have my template DNA that has those polymerase sites on each side. So you should have, if you're ready to do riboprobe synthesis, you should have already taken your plasmids and amplified rather the insert of those plasmids, and then purified that with the Zymo kit. So if you have that, that's your starting material. So if you're on your in vitro synthesis of riboprobes protocol, the first steps I've already completed. That's using the hot start PCR to make the template, and running it out on a gel to check it, and purifying the PCR product with a Zymo kit and eluting in 20 microliters of water. And I actually use Dep-C treated water, RNase-free water is fine. And then the transcription reaction is what I'm going to set up now. This includes several components. It has a buffer, and this buffer will be different for your T3 reactions. You'll use 10x transcription buffer, looks like this. And for SB6, we're going to use this high yield transcription buffer, which looks like this. When you get these out of the freezer, you want to make sure that they are thawed completely, and also sometimes there's some DTT that will precipitate out, so you want to make sure any precipitate is fully resuspended before you use these buffers. The next thing this says is T3 or SP6 RNA polymerase. Those are our enzymes. So those are in the refrigerator, and we can only get those out in that cold block because they're enzymes. We want to keep them working well. The next is an RNTP mixture with 10 millimolar rGTP, rATP, and rCTP, and 6 millimolar rUTP. So if you haven't made this solution before, you're going to get out these RNTPs, and you're going to make a mix of those that has 10 millimolar or 6 millimolar of each RNTP in a 10-to-6 mixture. So if you haven't done that yet, you can do the calculation. Usually I just make 100 microliters of that at a time. You don't want to freeze and thaw this too much. The reason that there is less rUTP in that mixture is that we're also going to add, and the next component is DIG11UTP, which is in a tube like this. This is also in the freezer box. And DIG11UTP is a UTP that's been modified to have this small chemical called digoxygenin on it. And this is what's going to be recognized by an antibody in the in situ hybridization reaction. This is what allows us to actually examine the riboprobe and see where it is with downstream steps. So because we're using this Dig 11 labeled UTP, we want to add a little bit less of the regular UTP. And the reason why we add some regular UTP is that helps with processivity of the RNA polymerase. If you just added Dig 11 UTP, you'd have a hard time getting the polymerase to read through the entire transcript. So you mostly add regular UTP and then a little bit of this digoxygenated labeled UTP. The next thing on this list is RNasen, so that's RNase inhibitor. This is also an enzyme that's in the freezer, so we'll leave that in the freezer box. There's nuclease-free water, and then this template, which needs to be in this range of concentrations. So I have a set of templates. Some of them will require SP6 as an enzyme, and some of them will require T3 as an enzyme. So I've set up a series of tubes that say RIB for Riboprobe, and then my designation, so these are, A1 or 1 or whatever you want to say. I've also labeled on here the template, so this is going to be TH and the date. And at the top of these, I don't know if you can see it, I've given myself a little code, S for SP6 and T for T3. That way when I'm setting up several samples, I know which tube is going to get which enzyme and buffer and so on. And so I've calculated master mixes. I've written out all my probes that I'm going to make, and I have written out the same kind of reagents that are on the protocol and the amounts. And then I've multiplied this to give me a list of reagents for SP6 and T3. So of course the enzyme is going to differ between these. So the enzyme for SP6 is obviously going to be SP6 polymerase, and T3 is going to be T3 polymerase. The buffer is going to be 10x transcription buffer for T3 and high yield transcription buffer for SP6, and everything else will be the same between the two. So the first thing I'm going to do is make my master mixes. So I have two tubes, one just has T and one has S for my T3 master mix and my SP6 master mix. I'm going to first add the water. Kind of inconveniently, I have two different amounts or numbers of reactions. Make sure you don't mix these up. So I have SP6 on the left and T3 on the right, so that's the order I'm going to have my tubes in so that I don't get mixed up. After I add the water, I'm going to add the transcription buffer, and I'm going to make sure I have the right one. So my first tube is SP6, so I'm going to add the high yield transcription buffer for that one. And I should have mentioned, I'm using RNase-free filter tips for all of this pipetting, since this is RNA work. We're trying to make single-stranded RNA. This will actually be the trickiest synthesis reaction that you do in the lab, probably. Just because it's single-stranded, the double-stranded RNA that you might have made before is actually a little bit more stable. So I'm adding everything but the enzyme. So I've added the water, the buffer. I'm getting ready to add the RNTPs. So this is my 10-to-6 RNTP mix. And then I'm going to add my Dig 11 UTP. There's not a lot of this reagent in the tube, so you may need to spin it down to make sure you have enough. And you don't add very much of this to the reaction, so one tube of this should be able to go quite a long way. Okay. So the only thing I need to add still are my polymerases. I've kind of done a cooking show trick, and I have already gotten my T3, my SP6, and my RNase inhibitor out. I put them in the cold block and then just put the cold block right next to me in the freezer. So So this has been in the freezer the whole time, but just I put it close to me so you don't have to go all the way to the freezer. But the enzymes I have are T3 polymerase, SP6 polymerase. These are enzymes that we make in the lab, and then the RNase inhibitor, which will either have a pink lid. This is the one we have now, and occasionally we'll have a different colored lid if we get a different type. So at this point, I'm going to add the enzymes. Remember, you're adding T3 enzyme to your T3 master mix. And you're going to add SP6 enzyme to your SP6 master mix. And remember, we've talked about master mixes in other videos, but just in case you haven't seen that one yet or haven't thought about it in a while, we're making master mixes because it's quicker. You do less pipetting, it's fewer tips that you're going to have to add, and also reduces the rate of errors. So you're not pipetting, 0.4 microliters many times. The last thing I'm going to add to these mixes is the RNase inhibitor. I'm adding that to both tubes. And now, as we talked about in other videos, you want to make sure that your enzymes are back in the freezer. So I'm actually going to put back the enzymes, but also the DIG 11 UTP and the RNTPs, and then I will be back momentarily. Okay, so again, I've already labeled all of my tubes, and I've written at the top S or T so that I know which of my master mixes will go into each tube. I'm going to just take these two tubes and flick. You can also either flick them kind of with your wrist down or put them in the centrifuge briefly to get them all to the bottom. And then in this reaction, we need 14 microliters of the master mix for each tube. So I'm going to very carefully, I'm going to use do my SP6 tubes first, so all the ones that I've labeled with an S. You can actually, we talked about needing to make antisense riboprobes. You can make a sense riboprobe, and the prediction would be that a sense riboprobe would not give you any signal. So that's one kind of control you can do for in situ hybridization experiments. Okay, so that's all of my SP6 master mix. And now I have the T3 master mix. I'm just using one tip for all of those because I'm pipetting into empty tubes, and it's a master mix. But for many of those chemicals I was adding, I was just using a tip once, when I was making the master mixes. So once this, the master mix is distributed into each tube, I just have the tubes like this. I'm leaving their lids up. This is kind of a little trick to help you remember where you are. So I am going to open them as I add the master mix and then close them as I add the PCR. And just make sure you are paying attention to which tube gets which template. It's always important to kind of line them up carefully. Keep them in order as much as possible. Keep things simple. And so I'm just adding the template to each tube. I'll show you just one of them. I'm just pipetting right into that liquid and then stirring a little. As we said before, you don't really want to pipette up and down or vortex too vigorously when you have nucleic acids. That can cause shearing. And I'm still using the filter tips for all the steps. Everything in this whole protocol, you'll use the RNase-free tips and everything for. All right, so I'm finished with setting these up. I'm going to put them in the incubator, one of these little heat blocks at 37 degrees overnight. You can usually, in the protocol it says 37 degrees for 4 to 5 hours or 30 degrees overnight. We've been having pretty good luck with 37 overnight too. So you can kind of play around with this if you want to see what works best in your hands. So I'm just going to use one of these hot blocks, like this one right here, set it to 37 degrees and allow this to incubate overnight. Tomorrow we'll come in and finish the protocol. We'll do DNase treatment, precipitation, purification, and run a gel to check our probes. All right, so I've allowed these transcription reactions for the riboprobes to go overnight. We're on this in vitro synthesis of riboprobes protocol. And I've come in and I've taken my samples out of the incubator, and I'm ready to move on with the protocol. And the third part of this protocol says DNase treatment. So this protocol step is meant to eliminate the sample of the DNA that we put in for the template so that you only have the riboprobe present. If you were to skip this step, you would still have that template DNA around, and then when you tried to use your riboprobes in your in situ hybridization, what you might find is those riboprobes would bind to the DNA instead of the RNA target that you're attempting to visualize in situ hybridization. So in this step, we're going to incubate our samples with DNase enzyme. We're going to make a master mix, so we're going to combine these reagents for each sample. And I've already labeled my tubes. Master mix, riboprobe, that's the double strand one. I'm also doing something else at the same time. Master mix riboprobe T and master mix riboprobe S, because we're going to have a separate master mix for the SP6 generated probes and the T3 generated probes. And in my lab notebook page, I've already done the math to multiply these reagents by the number of samples I have so that I can combine them and then distribute them into the tube. So I'm going to add the water first, then the appropriate buffer, so either the 10x transcription buffer for the T3 reactions or the high yield transcription buffer for the SP6 reactions. I've already gotten these out of the freezer, made sure they were thawed and resuspended well. So the first thing I'm going to do is add the water. And I'm adding depth C water, so this is RNase free. We don't want those RNases to be degrading our riboprobes that we've worked so hard to generate. And I have some T3 reactions and some SP6 reactions, so I'm now adding water to my second tube. And then I'm adding a smaller volume of the appropriate buffer, so I'm adding high yield transcription buffer into my master mix for the SP6 probes. That was the buffer that we used with that enzyme. And then I'm going to add the 10x transcription buffer as the buffer for the T3 probe DNase treatment. And I'm going to briefly pause this so that I can go get the DNase from the freezer. This is an enzyme, so we're going to keep it on the cold block as we do with all the enzymes, and I'll be right back. So I brought back my cold block with my enzyme. This is they'll say RQ1 DNase on the lid. It's a promega DNase. And so now I'm just going to uncap that so I can add the DNase to each reaction, or master mix rather. Okay, so I've added the DNase because this is an enzyme. I want to cap it right away and take it back to the freezer before I move on. Okay, I now have 2 master mixes, one for each SP6 and T3. I'm just going to flick them a little to mix and snap my wrist to get them down at the bottom. If you need to, you can also centrifuge them briefly to get all the liquid to the bottom. And then you're going to add 5 microliters of the master mix to each tube. You'll recall that I have already put a little S or T at the top of my tubes so that I know which of my riboprobes I generated with SP6 or T3 without having to look at my notes and cross-check and things like that. So what I'm actually going to do is make 2 rows. So I'm going to put all of the SP6 probes in one row and all of the T3 probes in another row. I'm going to add the master mix for the SP6. I'm also just going to kind of stir them around with my tip a little. If you notice that your riboprobe synthesis reactions are not all at the bottom of the tubes, for example, if you kind of jostled them a little bit while you were moving them around, you might decide that you need to also stir, but also maybe centrifuge down these tubes. What you don't want to happen is for there to be droplets of your riboprobe synthesis reaction on the side of the tube, in which case the DNase would not access those and you would still have DNA present in your sample. So here's a good example where there's like a droplet there on the side of the tube, or maybe, you know, I flicked that tube a little bit. So what I'll do is just pulse centrifuge these down to make sure that the enzyme is in the right spot. So that's all of my SP6 reactions. Now I'm doing the T3 master mix, putting it into each tube. I'm doing a little stir. Again, this is a nucleic acid. You don't want to vortex. You don't want to pipette up and down too vigorously because you can shear the riboprobes and then you won't have the product of the reaction that you worked so hard for. So the master mix tubes can just go into the trash. All of our tips were in the tip box. I should have mentioned again, for all of this riboprobe synthesis, we're using the filter tips that are RNase free. And I'm going to put these back in the incubator at 37 degrees for 15 minutes and then take them out for precipitation. Alright, so these riboprobes have been DNase treated. And next we're going to do the cleanup by ammonium acetate precipitation. We're going to add these components just in the order that they're listed, and this will precipitate our RNA so that we can purify it and get rid of the enzymes and other materials that are in our tubes. So I have several samples. I'm just going to do the first 2 so that you can see what it looks like. And then I will do the rest off video so that you don't have to watch me pipette for too long. So the first thing we're going to add is 80 microliters RNase-free water. This is just to bring up the solution in a little bit more water. It's kind of a smaller volume. Okay. The next 2 steps are optional. I'm going to do them. So the first is to add 2 microliters of total RNA to each tube. So I'm just going to add them. This will actually, you can do a number of things. You can just add 2 microliters of just straight total RNA, and this is yeast RNA. What this is just regular RNA that can be used to be kind of as a carrier for your riboprobe. So it will mean that you get a larger pellet, and it's a little bit easier to execute the purification. I will say that because you've added this RNA, you won't then be able to quantify your RNA with a spectrophotometer because so much of the RNA will be from the yeast RNA, but that's okay. We're going to run it on a gel to check it out anyway. So I'm actually going to use a little bit less than is recommended. So I have a 1 to 10 dilution of the total RNA, and I'm going to add 5 microliters, but you can just do it the way that it's in the protocol if you want. I'm just trying to have there be a little bit less of that total RNA. And then I'm going to add the EDTA, and I've made this solution, but I have just a little tube of it. And I'm just kind of doing that swirl thing to pipette. Again, you don't want to pipette up and down or vortex. So after I've added those things, I'm going to add 100 microliters of 5 molar ammonium acetate. If you haven't already made this, you can probably get some from someone, but there's also a how-to for this specific chemical in the chemical preparation videos. This is a salt that's going to cause precipitation of nucleic acids in the solution. I'm adding these all with the RNase-free filter tips. And you might see that there's some kind of, I don't know if you can see it, kind of swirling as this precipitates. And then the last thing you're going to add is 100% ethanol. You're going to add 800 microliters. And the ethanol will also precipitate the RNA. You can change your tips each time if you think you might have contaminated, or you can use one if you don't touch anything. So I'm just going to invert these a few times. You may or may not see much of a precipitate. If you're doing this first, you probably won't get too worried about this, but if you've done the double-stranded RNA purifications first, you might find that this is a little bit stressful because the riboprobes, there's so much less of them than the double-stranded RNA that you see less precipitate both now and when you spin your samples down. So just be aware of that. Okay, so I'm going to add all the rest of the chemicals, but you don't have to watch me do all that. I'm just going to do the same thing with all my other samples. I'm going to incubate these samples at -20 for at least 1 hour or overnight. So if you needed to do this and then get back to it after the weekend, that's fine too. But it needs to be at least for an hour, and the cold helps to precipitate the RNA. So often I'll do a little bit over an hour if I can, but an hour should be sufficient. During this time, I'm going to turn on the centrifuge and turn it to a cold temperature. That way, when I'm ready to start spinning down the riboprobes, the centrifuge will already be cold and I don't have to wait 15 minutes or whatever for the next step. So that's it for now. I'm going to finish with these and then put them in the freezer for an hour. So after my samples have precipitated for over an hour, and I let mine go a little bit longer because I was just doing something else, that's fine. I'm going to take my samples out of the freezer and put them directly into the centrifuge. And the centrifuge has been pre-chilled, so it's cold, the rotor's cold and everything. And I am going to allow these samples to spin, and this will take that precipitated RNA and pull it down to the bottom of the tube in a pellet. And to do this spin, we are going to set like I said, we've set the temperature at 4 degrees, so the centrifuge is already cold. The first spin we're going to make is 15 minutes at max speed, which on the centrifuge is a little over 20,000 Gs. So we're just going to hit start there and allow this to spin for 15 minutes. If you want to run your samples out on a gel when you're finished, you can take this 15-minute spin to pour a gel. There's a separate video about how to pour and run an agarose gel if you haven't already done that. And so you can set up that gel and it will be ready for you once your samples are ready and purified so that you can check them out. I realized that I wanted to mention one specific thing about centrifuging these samples. Because these pellets are going to be very small, I always put the hinge of the tube facing the outside of the centrifuge, so on the outer wall of the centrifuge. This means that the pellet will end up on the same side as the hinge, so it'll be easier to find. If your tubes are just sort of randomly distributed, then you're not sure exactly where to look for these tiny little pellets. So I've pulled out these samples. Sometimes you can see a faint little pellet. Sometimes you can't see much of one. That doesn't mean that there's not one there. There's some little tiny pellets there, but I'm not sure you can see on the camera. What you're going to do either way, if you see one or if you don't see one very well, is remove the liquid starting on the front wall, so the non-hinge wall, taking out the liquid like this. That way you don't suck up that pellet, and you're just kind of gently taking the solution off. In this first pipetting, you're going to use a different tip for each sample, and you're also not going to remove all of the liquid. You don't have to totally worry. You don't want to knock that pellet off, so I just gently get as much out as I can on the first try. And then what you'll do, I'm going to just do it for the first 2 tubes, is add 500 microliters of 70% ethanol, and this is 70% ethanol made up with Depsy water. So I'll just do this for the first couple so you can see what it looks like. And this 70% ethanol is just going to wash those pellets. And sometimes you'll notice that your pellets turn a little bit whiter and are easier to see once they're in 70% ethanol. So this one I couldn't see the pellet very much before I added the 70% ethanol, but it turned a little bit whiter with the ethanol. So I'm going to spin these samples again when I finish doing this for the rest of my samples at the highest speed at 4 degrees, but only for 5 minutes. And then we'll be back and finish up. Okay, so these Riboprobes have been spun for the second time. I'm not sure if you can see the little pellets there. It's just right there, very tiny. And so what I'm going to do is I'm going to take my P1000 pipetter, which I use to add 500 microliters. I'm going to set it up more closer to 800. And I'm going to remove as much of this liquid as I can. Then what I'm going to do is take these tubes, once I'm finished, and spin them down, just a pulse centrifuge. It can even be one of the tiny centrifuges. And then I will come back with a P10 and also the filter tips and remove the rest of that supernatant. So I'm going to remove the first bit for all of them. I'm centrifuging, that's why it's like a little loud. So again, I'm going to just remove the first bulk of the liquid with a P1000. If you can't see the pellet, just try to do your best to avoid that back wall where the hinge is, but hopefully you'll be able to see them. And I'm going to do this for the rest of the sample, spin them down, and be right back. So after you've done your spin, you'll get a little bit more of the liquid from the tube down to the bottom. And then you'll take a P10 or a P50 or something like that has a finer tip, and you'll go down very carefully trying to avoid sucking up this pellet if you can see it. And it might take you a couple of tries to get all the liquid out, and you can re-centrifuge it in the middle if you need to. So if you can't see a pellet, you might be kind of worried by now. But occasionally the pellets are just so small they're not very visible. And especially if you've done, if you've made double-stranded RNA before this and you've seen the very large pellets that result from that reaction, this will be a little bit unnerving. But these pellets are very small, If you need to redo your reactions after this, you can add a little bit more of that yeast RNA so that you can see the pellet better, so that you can avoid losing it during the centrifugation steps. And so after I've removed the liquid from all of these, I'm going to let them sit on my bench for 5 minutes, just covered in a chem wipe, just kind of like this, so that they can dry and all that ethanol can evaporate. And when that 5 minutes are up, we'll be back to resuspend the Riboprobes. So after 5 minutes have elapsed, you can sometimes see the pellets at the bottom. You're going to add 20 microliters of Riboprobe resuspension buffer, or if you'd rather, sometimes we resuspend these in water or even in hybridization buffer. So there's a few ways you can do this. You can ask someone if you're not exactly sure. Depending on the size of your pellets, you can resuspend in different volumes. Make sure I don't miss one here. So in the protocol, it says resuspend in 20 microliters of water. You can also use this Riboprobe resuspension buffer. And there's a recipe for that on the bottom. And that can just be put in the freezer between uses, so you don't have to remake that every time you need to resuspend some probes. So then I would just kind of flick these, and I would let them sit for, I don't know, at least 10 or 15 minutes to resuspend, and then flick and mix well again. RNA sometimes take a little while to go back into solution, and so that will allow them to go into solution before you load your gel. Okay, once your Riboprobes have resuspended adequately, you're going to load them into a gel. And in the loading of the gel, I showed you how to make these little dimples and make little wells for your samples. What you're going to do is combine 6 microliters of RNase-free water, 1 microliter of the formaldehyde load dye, which is in the freezer. You can take it out and thaw it. It's not an enzyme, so it doesn't need to stay on ice. You can just pull it out as long as it goes right back. And then 3 microliters of your Riboprobe. If you have really strong pellets, you might only load 1 or 1.5 microliters. That would save you some so that you have more Riboprobe left over. But if mine were a little bit small today, so I'm going to load 3 microliters. And you're going to run this just a 1% agarose gel. Make sure you have the gel red in there. And what you're really looking for on this gel is just to see a band at all. So if you have a strong band or a weak band, either one is okay. They can usually both work. The worrisome thing would be if you ran this gel and got no band present, in which case I would probably start back at the Riboprobe synthesis step, so you don't have to necessarily redo the PCR, but I would probably double check that your PCR product looked good, and if it did, then start with the Riboprobe synthesis again.